NASA announces discovery of life on Mars with high degree of confidence. A recent analysis of the Sapphire Canyon mudstone core, drilled by NASA's Perseverance rover in July 2024, adds new evidence to the ongoing search for life on Mars. The study describes minerals and textures that, on Earth, are often linked to microbial activity. At the same time, the authors stress that non-biological chemistry could also explain the signals. This finding by Perseverance is the closest we have ever come to discovering life on Mars. The identification of a potential biosignature on the red planet is a groundbreaking discovery, and one that will advance our understanding of Mars, said acting NASA Administrator Sean Duffy. NASA's commitment to conducting gold standard science will continue as we pursue our goal of putting American boots on Mars' rocky soil. The core was taken from a rock named Shavaya Falls in Neretva Vallis, an ancient river channel about a quarter-mile wide that once fed Jezero Craters Lake. After drilling, Perseverance sealed the sample for possible return to Earth, where laboratory instruments can perform tests far beyond the rover's onboard capabilities. Inside the Martian mudstone, lead author Joel A. Hurowitz of Stony Brook University reports a fine-grained mudstone with circular reaction fronts informally called leopard spots, plus small nodules embedded in layered sediments. Perseverance's Sherlock and PIX instruments mapped organic carbon with phosphate, iron, and sulfur arranged in distinct, repeating patterns. Two minerals stand out: vivianite and greygite. Vivianite is an iron phosphate, while greygite is an iron sulfide associated with iron and sulfur cycling in oxygen-poor settings. These features show up in rocks that settled from water, not in lavas. The site on Mars lies along Bright Angel, a set of outcrops that preserve layers and veins consistent with slow changes after the mud was laid down. Textures and chemistry point to low-temperature reactions that reorganized elements already present in the mud. That detail matters because low temperatures fit environments that life can handle, while very hot conditions tend to erase delicate signals. Earth microbes leave similar traces on Earth. Vivianite often forms where microbes reduce iron in water-rich sediments and trap phosphorus in blue-green nodules. Laboratory and field work document biologically mediated vivianite through extracellular electron transfer. Greygite frequently appears where sulfate-reducing bacteria drive chemistry in anoxic mud. In controlled experiments, greygite was detected only in live biotic setups after months of incubation. The Martian rock shows rims rich in vivianite surrounding small cores enriched in greygite. That bullseye pattern matches a sequence of electron transfer reactions seen in some Earth sediment. None of this proves metabolism happened in Bright Angel mud, but it shows the chemistry is right for it. That is a subtle point, and it is the reason scientists keep the language cautious. Evidence of life on Mars, a potential biosignature, is a feature that might have a biological origin but still needs more data to rule out non-biological sources.